Now we're going to discuss the control of metabolism. When you think about all the different enzymes in a single cell, they don't always produce reactions the same time as every other. And at current estimate, we have close to 20 to 40,000 different proteins inside each cell, or the capability to produce that many. And of them, maybe half of them or more would be involved in metabolism. So how can all of those different reactions happen at the right time, at the right speed, and at the right instance for life to continue or to exist? We can think of each enzyme as a little machine or a little component of a metabolic machine. And each enzyme has a very specific function or job in that very complex process. By controlling the enzymes, then, whatever reactions each enzyme is responsible for can be controlled, and therefore you can ultimately control the entire metabolic reaction if you can find a way to control the enzymes. But what is control? We can think of control as two states. Something is turned on or something is turned off. You activate it or you deactivate it. In that case, you have control over something because you can decide or indicate when you want something to occur. But not everything that we're familiar with when you control it is in a state of just on or off. It's not all the way on or all the way off. Lights can be on and off, but some can be dimmed. If you have a dimmer switch, you can have all variations between on and off. So in other words, rate of reaction or speed of reaction is another important method of control. So dimming the lights means that you're somewhere between on and off instead of just on or just off. And in metabolic reactions, being able to control how fast a reaction occurs sometimes is just as important or more than if the reaction is on or off. So how is this done? How can 20,000 or more tiny little microscopic molecules be controlled by a cell which doesn't have any kind of cognitive abilities or brain or decision-making ability? And if these reactions were to occur at the wrong time, it would definitely result in the death or the inefficient operation of the cell. The answer then comes in this very simple sentence, which has a lot to it. Affecting the concentration of active enzymes is the secret to how a cell can control every metabolic reaction. The rest of this presentation is going to go into detail as to how this can happen automatically by the molecules themselves. The first of these different categories of control I call create and destroy. The create-destroy method of controlling metabolism involves the DNA of the cell, and it involves specific portions of the DNA referred to as genes, or instructions for how to make certain proteins or enzymes. By activating a gene, you instruct the cell to produce a particular enzyme or protein. In order to make more than one protein, the gene must be activated long enough to produce multiple messages to the cytoplasm. So, the longer that a gene is activated, the more enzymes of that particular type will be produced, and the more of that reaction will occur in the cell. In some cases, once the reactions begin to occur in the cell, the only way to get them to cease is to go out and destroy the same enzymes you just spent that effort making. So, to do that, in some cases, the cell actually produces a separate enzyme to go out and seek and destroy the original enzyme, and once the original enzymes are destroyed, the reaction slows down or stops. This, of course, if you imagine, requires lots and lots of energy to do. For every reaction, you may require millions of enzymes, and you need to spend the energy in a cell to make each and every enzyme. And to slow down or stop the reaction, you'd have to build another enzyme to go out and find and destroy the original. This would also result in a very, very slow response. The speed of getting the reactions to begin and the speed of turning off the desired reaction would take a lot of time. If we look at this, it would seem that why would a cell do something in such an inefficient manner? And the actual thing we should focus here on is what is the benefit of making it difficult for something to occur. Inefficiency isn't necessarily a bad thing when it comes to having control over something. Suppose a cell has a very dangerous or destructive reaction that it doesn't want to accidentally occur. Let's say apoptosis, which we discussed. That's where the cell initiates a self-destruct or suicide order for itself. You don't want your cells to just accidentally digest and kill themselves. In those cases, the enzymes that are responsible for that kind of a reaction or response, they shouldn't even be present in the cell until you know for sure that that cell is ready or destined to commit suicide. For the rest of our body cells, we may have the genetic instructions, but the genes are not activated. Therefore, the enzymes are not present, and the chances of accidentally killing a cell by suicide is 0. So that's one of the reasons why create-destroy is one of the methods of metabolism control. Now we can look at something else that's more practical. Another method for controlling enzyme activity is to change the reaction environment. So here RxN refers to as a shorthand for reaction. 3 typical ways can be utilized to change how quickly or whether or not an enzyme can function. And that is to alter the environment, the inside of the cell that surrounds the enzymes. Temperature is one of the most recognizable ways. If we were to increase the temperature way above or way below the functioning range of an enzyme, we would end up denaturing it. Too high a temperature, and we would end up breaking little fragile bonds like hydrogen or ionic bonds, and then the protein would start to unravel. An enzyme depends on its shape in order to function. And once those internal bonds begin to break and the protein denatures, then the functionality of the enzyme is lost. pH has the same effect. Too high or too low a hydrogen or hydroxide ion concentration, and internal bonds are affected and the protein could denature. Also, the concentration of ions in the environment, that's complete positive or negative ions. And these ions could then ultimately affect the hydrogen and ionic bonds as well, just like temperature and pH. For each of these 3 environmental factors, there is an optimum. So if you look here on the graph, how fast an enzyme works or how quickly a reaction occurs depends on what the temperature, pH, or ion concentration is. At one specific temperature, you'll notice that the reaction rate is the best or the highest. Anything above or below that temperature, in this case it looks something like 42, 43 degrees Celsius, then the activity of the reaction of the enzyme drops off. At this point above this temperature, the enzyme is denaturing. Below this, it could be a combination of denaturing or just slower collisions of substrate with the enzyme's active site. Either way, in order for the enzyme to work at its highest speed, you must have the environment of the cell at the perfect temperature. So inside the body, why are we concerned with 98.6 degrees? Because that is the optimum temperature for the majority of our enzymes. We also have an optimum pH of bodily fluids, and we have an optimum ion concentration. All three of these must be at the optimum level for the enzyme to work its fastest. And decreasing any of these other factors can affect how fast the enzyme works. So it relies on the environment being perfect in all three ways. Now there's a negative effect of trying to control an enzyme using this method. The negative effect is that all the enzymes in that environment are affected by the change in temperature, pH, or ion concentration. So this is not a really good way to change how one enzyme works among a lot of others. On the other hand, there is a positive. Suppose you want all reactions to cease or not occur except for one or two. In this case, changing the environment makes it a bad place for a lot of enzymes except for the one that's specially able to tolerate those conditions. So in class I mentioned the example of digestion in the stomach. In the stomach we do not want any reactions occurring, so the pH of 1 that's produced by the hydrochloric acid ceases and stops all metabolic reactions. So any food particles or organisms you might consume, their metabolic functions cease. But the only one that is allowed to occur is protein digestion by the enzyme pepsin. pH 1 happens to be the perfect pH for the digestive enzyme pepsin. So in that case, the stomach is going to be the best environment for digestion of proteins to occur. But what would happen if the stomach contents were moved to any other part of the body? Well, the pH 1 environment is only in the stomach. So once the food moves on to the small intestines, where the pH is raised well above 1, the digestive enzymes stop working. Otherwise, you would actually digest yourself, starting with your small intestines.